we can get started. Hi, everyone. Welcome to the UW Division of Rheumatology Ground Rounds. Just a couple of reminders. This session is currently being recorded and will be posted later on our website. There will be a Q&A session at the end, so please add your questions in the Q&A feature and not the chat box so that it's easier to moderate. And now I'll let Dr. Elkon introduce our speaker for today.
Okay, good morning. I'm pleased to introduce Ann Davidson to give a grand rounds. She obtained her medical degree in Melbourne, Australia, and did her fellowship at the Albert Einstein College of Medicine in New York. She's currently professor in the Department of Molecular Medicine at Hofstra Northwell in Long Island. By way of background, the overarching themes of Dr. Davidson's research have been the investigation of autoreactive B cells and studies of lupus nephritis. Following initial contributions to understanding anti-DNA idiotypes while still a fellow in Betty Diamond's lab, she went on to explore the roles of co-stimulatory molecules as well as the B cell maturation protein BAF in adaptive autoimmune response. And has also pioneered therapeutic strategies to block these pathways in mouse models of lupus. More recent areas of her research concern the pathologic role of macrophages in lupus nephritis. These studies have included definition of myeloid phenotypes and transcriptional profiles in murine lupus kidneys. Recently, Dr. Davidson joined the AMP consortium and applied her considerable body of knowledge of myeloid cells in murine lupus to human lupus nephritis. And these have considerably furthered our understanding of the nature and pathogenic potential of myeloid cells in the lupus kidney and urine. And that's what she's going to be talking about. Just to mention that Dr. Davidson has received prestigious awards from the ACR, the Dubois Award, the Distinguished Investigator Award, and recently became an ACR master. So, and we look forward to hearing what you and the AMP are up to.
Okay, great. Thanks very much. I can't see anyone, so I'm just going to assume that everybody is there. Today, I'm going to talk about our work as part of the Accelerating Medicines partnership, as well as some collaborative work I've been doing with scientists at the Broad Institute. So I'm going to divide the talk into three parts. First, I'm going to give a brief primer on lupus nephritis. Then I'm going to give a high-level view of single-cell RNA sequencing data from the AMP. And then I'm going to talk about our work in mouse models to discover new myeloid cell subsets and functions. So I'm going to start with a very brief primer. I think everyone here is familiar with lupus as being an autoimmune disease in which genetic and environmental triggers will initiate both an adaptive and an innate immune response, which becomes dysregulated, resulting in the formation of autoantibodies and immune complexes. And these then cause inflammation and tissue damage. And then this results in the release of soluble mediators, lots of amplification feedback loops that then amplify the whole process. So today we're really talking more about tissue injury, and tissue injury is what leads to chronicity of disease, damage, and decreased quality of life. So again, starting with these abnormal immune responses and amplification loops. In the tissues themselves, there are metabolic disturbances, inflammatory cell recruitment, loss of homeostatic programs, dysregulated repair, and finally, loss of cells and structure and fibrosis. And these latter things start to become irreversible. And today we're talking about the kidneys, although lupus involves many other organs apart from the kidneys, as you all know. And our goal really in lupus is to prevent this irreversible damage in our patients. In the initial stages of lupus, a lot of what we see in our patients is reversible, but in the chronic phases, it is not. Okay, so lupus nephritis. So again, talking about initiation of disease, lupus nephritis is initiated for the most part by deposition of immune complexes in the glomeruli. And in the glomeruli, I'm not sure if you can see my, my pointer, these immune complexes will deposit on either side of the glomerular basement membrane shown here, and then they will initiate inflammation with recruitment primarily of macrophages and T cells in the glomeruli. This results in inflammation and recruitment of inflammatory cells that gather around the outside of the glomeruli, as shown here. And then because the blood supply of the tubular interstitial space derives from runoff from the glomeruli, glomerular hypertension and glomerular dropout will result in less blood supply to the tubular interstitium, resulting in tubular interstitial hypoxia, tubular damage, and this then will result in recruitment of interstitial infiltrates into the tubular interstitium and induction of fibrosis. Okay, let's just... Okay, so renal hypoxia is something that we worry about a lot in patients with lupus nephritis, and our patients have lots of risk factors in addition to inflammation for renal hypoxia, and these include anemia and hypertension, atherosclerosis, smoking, hypercholesterolemia, and previous episodes of renal injury. Initially, renal hypoxia will result in the induction of protective mechanisms. So these include increased secretion of VEGF to protect the capillaries, stabilization of HIF-1 alpha, production of MMFs to break down any initial fibrosis, and increased antioxidants. But if the renal hypoxia continues, these mechanisms become dysregulated, and now we start to see a decrease in VEGF, loss of the capillaries, a decrease in HIF-1 alpha, apoptosis of epithelial cells, recruitment of fibroblasts, local inflammatory cells, and lots of abnormal gene expression. So it's been shown by Joe Kraft that renal hypoxia contributes to interstitial inflammatory cell recruitment. And these were studies he published several years ago now, in which he showed that in areas where there's expression of HIF-1 alpha, there's also recruitment locally of interstitial T cells. And these interstitial T cells are abnormal in that they upregulate HIF-1 alpha, and they stain for a hypoxia marker. Okay, so let's move now to how we manage our patients and what we can learn from their kidney biopsies. So kidney biopsies are really the gold standard for diagnosis of lupus, and we use them to make a plan of treatment. However, the classification of the histologic class on the renal biopsy does not very well predict either the prognosis or the response to treatment. And so we measure an activity score, which will drive our treatment decisions, but just looking at the activity score on the renal biopsy doesn't predict the outcome. What does predict the outcome are all the components of the chronicity score. So these are things to do with the more irreversible aspects of renal damage, including fibrosis and tubular atrophy. We also know there are demographic and clinical factors associated with poor outcome, and these include age, which is actually being confirmed in the AMP study, duration of renal involvement before the biopsy, whether the patients have hypertension, whether or not they've got a high serum creatinine. So any evidence of damage at the time of biopsy is associated with a worse outcome. So repeat biopsies would be useful because they might be helpful to monitor the response to therapy and to make further treatment decisions, but they're very rarely performed. And even at repeat biopsy, the histologic findings may not entirely reflect the clinical state. So one can have ongoing renal damage without too much abnormality on the repeat biopsy, and conversely, somebody in remission may have a lot of interstitial infiltrates. So the question really is, can we do better than this? And this is what drove the beginnings of the AMP. And the goal of the AMP was to look at kidney biopsies from patients and to use lots of technologies to see if we could find either better predictors, or if we could understand the pathogenesis better. So I'm going to move now to the next part of the talk, which is a high-level view of the single-cell RNA sequencing data from the AMP. And I just want to acknowledge that the AMP is a very large team. It comes from a number of different centers shown on the left, and the leader at each center is highlighted in bold. And then it was supported by the NIH and by pharma in giving funds to the foundation for the NIH to support the AMP. So they've had a lot of input into general strategies as well. Okay, so what were the questions we were asking in the lupus part of the AMP? So the first question had to do with the molecular features of the kidneys. So could we understand what was going on in the kidneys by looking at single cells? And this included both the stroma of the kidney and the infiltrating cells. And if we did that, would it help in terms of either understanding prognosis or understanding therapies, or doing a better job than what we currently do with a kidney biopsy? One very important question here is whether there are pathways of disease we've been missing that require more mechanistic investigation, or that might require different therapeutic approaches, or might direct us to a personalized approach or a new approach. In addition to the single-cell RNA sequencing, there were a lot of other things that went on with the AMP, one of which is being undertaken by the Johns Hopkins group, being led by Andrea Fava. And there are a number of papers that he's already published on this, by trying to understand more about disease through examining proteomic profiles, predominantly of the urine. And you can look at his papers. He's got some very nice papers in this area. We were also asking whether either blood or urine could be used as a surrogate for a biopsy. So can we use urine as a liquid biopsy? In addition, there was some accessory types of investigations, such as, is it safe to perform a research biopsy? And that paper's been published, and the answer is yes. You can actually perform up to 4 passes before there starts to be an increased risk of bleeds or other adverse effects. There were some exploratory technologies used to begin to obtain some spatial data, and this is going to be done in the next iteration of the AMP, some comparison with other renal diseases, and we wanted to be able to use the data as a guide for further mechanistic studies in mice. Okay, so the clinical design of the AMP was very simple. The inclusion criteria, they had to have lupus, they had to be more than 18, their doctor had to have said they needed a kidney biopsy, and they needed a urine protein creatinine ratio of greater than 0.5. This decision of the UPCR cutoff required multiple meetings and discussions, which I won't bore you with, but it was a very contentious part of the decision. And then they needed to have lupus nephritis on their biopsy, but not class 6. So they couldn't have fibrotic kidneys, but they needed to have a diagnosis of lupus nephritis on their biopsy. There were a few exclusions, including kidney transplant and pregnancy. And then the patients were seen at baseline at 12 weeks, 26 weeks, and 52 weeks, and blood and urine was collected at 12, 26, and 52 weeks, as well as the biopsy at baseline. Okay, so just to point out, this is not a clinical trial. This is more like an observational, interventional type of study. And so the patients were allowed to be on whatever medications their doctor prescribed for them, and these were recorded at each visit. Okay, the definitions of remission are shown here. So a complete response was defined as a UPCR less than 0.5, a normal creatinine, and prednisone less than 10 milligrams. A partial response was defined as a greater than 50% reduction in the UPCR, normal creatinine and prednisone 15 milligrams or less, and non-response was a less than 50% improvement in the UPCR or failure to meet the other criteria of serum creatinine and prednisone. Okay, so just looking at our complete, partial, and non-response numbers in the AMP, there were about 250 patients enrolled. Not all of them were enrolled into the single cell studies. So if we look at complete remission rates, we see, as we've seen time and time again in multiple studies, complete remission rates of less than 30%, 28%, partial remission of 23%, non-response of 49% in these patients. And as a kind of sanity check, the complete remission rates were inversely correlated with the chronicity score on the renal biopsy. So if you had a chronicity score of greater than 5, you were very unlikely to have a complete remission. Okay, so the pipeline for the AMP data is shown here. Let's start with the kidneys. So patients were seen at the time of biopsy and the kidney was collected. Some of it was retained for histology and most of it went for single cell RNA sequencing. 155 patients had single cell RNA sequencing. We also had 30 healthy controls that came from healthy living transplant donors. A small portion of the kidneys was also used for single nuclear RNA sequencing. That was a subset of the patients. And the reason for doing this is that it had been reported that some of the stromal cells were better preserved if one did single nuclear RNA sequencing. So a subset of patients had this as well. Clinical data was collected, blood was sent for cytoph, urine was sent for single cell RNA sequencing, and urine was also sent for histology and for proteomics. And if we look at our 155 patients who had the single cell analyses done, they were mixed in their class. You can see about 35 to 40% had proliferative class 3 or class 4 disease, 30% had class 5 disease, and 30% had mixed proliferative and membranous disease. So a fairly good representation of lupus pathology. Okay, so there were some major challenges that we considered when doing these experiments. We knew that epithelial cells are very fragile. They die very quickly. They also have high levels of mitochondrial RNA, and we were worried that we would have difficulty in capturing some of the rare populations. And for this reason, we used single nuclear RNA-seq to make sure we captured everything. In the end, it turned out that we did capture all the relevant subtypes with our single cell RNA-seq, but this was not a given at the beginning. And then the decision was made to do all of the tissue disaggregations and library generations and everything else at a single site by a single set of operators. And this turned out to be important as well. Okay, so the pipeline for the kidney, we spent about a year optimizing cryopreservation protocols and found that cryopreserved biopsies, as long as they were cryopreserved quickly within the first 4 hours, actually worked very well. These were then dissociated, put on the 10x machine. And because there were a lot of mitochondrial reads, they were then processed with this technique called DASH, which was basically a way of removing all of the mitochondrial sequences so that the sequencing would allow more reads of transcripts. And after all this was done, the single cell data was accumulated. Okay, so we did learn a number of technical lessons here. And the first is that the cell dissociation of cryopreserved tissue actually produced a lot of high quality cells, including immune cells. And the background could be mitigated with DASH to cut down on the sequencing costs, as well as very careful QC. I'll show you that in a minute. The AMP approach was effective, so the samples collections were decentralized. And even after 4 years of cryopreservation, we still got pretty good high quality yields of cells. Fibrosis didn't prevent the cell dissociation, so even the fibrotic kidneys gave us samples. We found that 10,000 cells was optimal for loading of the 10x, and that the longer biopsies could be divided and used for both single cell and single nuclear cell RNA-seq. Okay, so the analysis workflow is shown here. And so the cutoffs were QC'd this way. So the number of genes had to be greater than 500, the number of UMIs greater than 1,000, and the percent mitochondrial reads less than 3%. And all the cells that expressed multiple markers of various cell types were removed. And then the data was normalized and PCA was performed on the 2,000 most variable genes. And then the data were harmonized using this program called Harmony, developed by Shomarad Chowdhury's lab. And then the cells were clustered and annotated. And again, anything that looked as if it expressed markers of more than one cell type was removed. And you have to remember that the nephron is extraordinarily complex, as shown here, that there are multiple types of tubules, multiple types of endothelial cells, not to mention all the complexity of the glomeruli and all of the infiltrating immune cells. Okay, so this required new analysis tools that were developed by the AMP bioinformatics group, particularly Shomarad Chowdhury's group. And one of the first things he developed was a program called Harmony, and that's shown here. So if you take cells, you take batches of cells and you just cluster them, they will cluster separately depending on the batch. But you can then take cells and find cells that are close to each other, and then you can try and push those closer together. So these cells here are likely to all be part of the same cluster, whereas these cells over here are unlikely to be. And this effect works extremely well. They published this method probably 2 years ago, and now everybody uses it for everything with a batch effect. The second thing they carried out was this neighborhood analysis. So this thing called covariant neighborhood analysis, again, they've published this, and this is a way to correlate neighborhoods of cells with clinical features without worrying what cluster they belong to necessarily. And then they used some various other things, including reference mapping to compare the cells with other tissues and other cell types to allow better identification of smaller subsets. Okay, so I'm first going to show you the immune cell clustering. There were 73,000 immune cells, and these were clustered based on their canonical markers and also compared to reference data sets. And then we looked at differentially expressed genes across the cell clusters. And you can see that the immune landscape was extraordinarily complex. So here I left the labeling in for the T cell clusters. You can see there are multiple T cell clusters, 25 clusters, and these could all be identified based on the reference mapping. And if we look at B cells, it's not quite as complicated. Plasma cells ended up being quite complex with lots of different clusters. And I will come back to the myeloid cells in the third part of the talk. There were 23 clusters in the initial clustering. I think we're at somewhere like 25 final clusters of myeloid cells. Okay, so one of the first questions we asked was how heterogeneous the patients are. And so just looking at heterogeneity of patients, and using principal component analysis, a lot of the variability could be explained by the first 4 principal components. And the first principal component had to do with the ratio of myeloid cells to T cells. So some patients had a lot of myeloid cells and fewer T cells, and other patients had lots of lymphoid aggregates and not so many myeloid cells. So in fact, this myeloid cell signature, having more myeloid cells, was actually associated with less chronicity, ability to respond to treatment, and a younger age. So you can look at it backwards. The less T cells they had, suggesting the less interstitial infiltrates they had, the less chronicity they had, and the more likely they were to respond to treatment. So the next PC is over here, and this had to do with how many B and T cells they had. So if they had more B cells, they tended to have higher disease activity. And then the third PC had to do with the T effector cells. So if they had high numbers of T effector cells, this was a little also counterintuitive. They were more likely to respond to treatment. And finally, and I'll come back to this, if they had a lot of differentiated monocytes, so that's in this cluster here, and I'll come back to this, and they had lower numbers of blood monocytes in their kidneys, this was associated with higher disease activity. Okay, so I just want to mention briefly that the AMP really is looking at frequency of cell types and not looking at numbers of cell types, because it's harder to do that in a very small biopsy of the kidney. And it's not looking at where the cell types are placed in the kidney. So that's being done by other components of the AMP. So this is just an example of a covariant neighborhood analysis looking at the myeloid cell clusters. And so if it's red, it means it's associated with whatever it is that you've asked it to look at. And if it's blue, it means it's negatively correlated with that variable. So one of the first things we saw was that there were a macrophage subset that was highly associated with the histologic activity score. That's shown here. And then if we looked at chronicity, it was a different set of myeloid cells that was associated with chronicity down here, as well as a subset of the cells that were associated with activity. So of course, we've been very interested in what these cells are and whether or not we can see them. And I'll just show you an example of this because we don't have all the data yet. So this is a single biopsy sample showing 2 glomeruli. And what you can see is the first glomerulus is a reasonable size, and it kind of looks okay. And it doesn't have too many immune cells identified by CD45 in brown. It doesn't have too many immune cells around the outside. And this is staining with 30 different markers, and I can't show you what they all are. And this is a very damaged glomerulus. You can see it's small. It's sclerotic, actually, when you look at H&E. And now you can see around this particular glomerulus, you've got a lot of immune cells have gathered around this particular one. And you can see here some of the subtypes of these immune cells. So what I'm showing here is glomerular staining with two macrophage markers, CD68 and CD163, and you can see that they stain different cells. And now if we look at these two subsets of macrophages up here, and these are macrophages that we know have a signature of phagocytosis, that these phagocytic macrophages, and now I'm showing you in situ hybridization of markers expressed by these cells, these are located inside glomeruli. We can see them here. But now if we look at these macrophages over here, these ones down here are dendritic cells, so I'm not going to talk about those, but these macrophages down here, these are resident macrophages, and these are all in the interstitium, you can see here. So we're starting to be able to do some visualization, and I'm showing you this just to show you that it's possible to do it. This will be done in much more detail by the next iteration of the AMP. Okay, so let's go back to the AMP, and I'm going to show you now the non-immune cells. So there were 163,000 proximal tubular cells. These are distal convoluted tubule. These are various other stromal, and these are ascending thin limb and thick ascending limb. And you can see that there are multiple clusters of these as well. Some of these associated with chronicity. So if we look at the proximal tubular cells and we look at correlation with chronicity, there was a repairing tubular signature that associated with chronicity. There was also a repairing signature of the ascending thin limb that associated with chronicity. And there was some association of the distal tubule as well with chronicity. So what does this actually mean? So we know that tubules are easily injured. They don't have a good blood supply, and they're easily injured by hypoxic injury. And when that happens, they can do one of two things. They can either redifferentiate and regenerate and look more or less normal again, or they can undergo this kind of dysregulated pathway of maladaptive repair, and then this will recruit inflammatory cells, because these will start to secrete inflammatory factors, and then this will bring in myofibroblasts and will induce fibrosis. So what we could see in the tubular cells, and I'm just showing proximal tubular cells here, is that there were a number of different clusters of proximal tubular cells, and the ones that I'm showing here were ones that had dysregulated metabolism, so they had a signature of dysregulated metabolism, and there was also a signature of dysregulated repair. So these ones here are more healthy looking tubular cells, and their metabolic genes are downregulated in these 4 clusters that have the maladaptive repair, And these clusters with maladaptive repair also express some genes associated with maladaptive repair. So these would be kind of healthier looking clusters, and these ones with the blue dots on the bottom are less healthy looking clusters. So do these associate with lupus nephritis? And they do, in fact. And if you look at the proximal tubular cell clusters, there were a number of clusters that associated in a case control study with lupus compared with healthy control. So we can see particularly this cluster here associated with lupus rather than with being healthy, and whereas this cluster here associated with being healthy. So if we look at these clusters, what we see is that the lupus-associated clusters express a number of genes that are associated with maladaptive repair. So these include things like keratins, vimentin, and this transcription factor SOX4. And SOX4 actually drives a redifferentiation program and epithelial development program. And this is mouse data, but the mouse and human programs are very similar. So this looks like it might be a dedifferentiation type of program that's going on in the lupus patients. Okay, just one quick word about having to adjust for confounders. All of the data had to be adjusted for confounders, and the things that we adjusted for included batch, race, ethnicity, prednisone use, activity and chronicity, sex, and responder status. So all of that had to be incorporated into these various models, and I don't have time to show you all of that data. So the conclusions from this second part are that we can get high-level single-cell data from cryopreserved samples, that using unbiased bioinformatics approaches, we can now start to address correlations of various aspects of lupus nephritis with gene expression data, that the data addresses cell frequencies, but not necessarily spatial organization or function. And we really were not able to define discrete subsets of patients, but there was some dichotomy between patients with a lot of myeloid cells versus lymphoid cells, and there were several features associated with worse outcome. Okay, so what do we do next? So spatial transcriptomics and proteomics is going to be done in the next iteration of the AMP. Organoids would be very nice, but are still rather primitive when it comes to kidneys because you can't really get the immune cells in there. And that's a problem for studying immune stromal interactions. And so we have, for a long time, wanted to work in appropriate mouse models. So I'm going to show you data that comes from a collaboration that I've had over the last number of years with the lab of Neha Cohen at the Broad Institute. So mouse models of lupus nephritis are very useful, but they're very heterogeneous. And so some of them, like the ones shown in red here, have a lot of inflammation in their kidneys. You can see all the blue here. Some of them overexpress toll-like receptor 7. That's these two green mouse models here. This particular one has very little interstitial inflammation, but has a lot of glomerulosclerosis. And then they're characterized by different autoantibodies, different gender, and different types of infiltrating cells. So it's hard to know what mouse model to use to model humans, and maybe we need to use all of them. So in very old data that came from my lab, we started with 3 different mouse models, and we did microarrays at that time, and we overlapped our microarray data with human microarray data that had come from the lab of Matthias Kretzler at Michigan. And what we found was that all of the mouse models and humans had a signature that had to do with dendritic cell and macrophage activation, as well as fibrosis. And this was the beginning of when we became very interested in these cells. And when we went back and looked at the literature, indeed, there was very nice data showing that in renal biopsies from humans, if those patients who had a lot of macrophages in their kidneys had a poor outcome of their nephritis and lost their kidneys within 10 years, whereas those who did not have macrophages in their kidneys did much better. And this was confirmed using data from Marcus Clark's lab, where he showed, and he divided the patients into 4 quartiles, depending on how many interstitial infiltrates they had. And the patients in the 4th quartile who had the most infiltrates did the worst over time. So this suggested that there was something that was pathogenic about these cells. So what kind of cells are in the kidneys? So it turns out that the myeloid cell population in the kidneys is very heterogeneous. And if you look at myeloid cell differentiation, so this is a differentiation map, all the cells outlined in yellow can be found in the kidneys. So these include plasmacytoid dendritic cells, shown here, resident macrophages that come from the yolk sac macrophages, shown here, various types of dendritic cells, shown here, and infiltrating monocytes, shown here. And these 3 cell types, resident macrophages, monocyte-derived DCs, and CDC2s, were really difficult to separate from each other just using standard flow cytometry. We can do a better job now, as I'll show you, but they were very difficult to differentiate. So I just want to point out that resident macrophages in normal kidneys are very plentiful. And here in a stain showing tubules, you can see that they're abutting the tubules very nicely. And here in a stain showing endothelium, they're abutting the endothelium. And these resident macrophages in high power view are very, very close to endothelial cells. Endothelial cells are in purple, and tubular cells shown down here. There is a basement membrane between the tubule and the resident macrophage cells shown here, but there's no basement membrane separating these 2 cell types in the kidneys. And in fact, the macrophages can eat immune complexes that come from the blood very happily. And this just shows you where dendritic cells are. There are a few of them scattered in the interstitium, and they're actually associated with collagen bundles shown in blue. Okay, so we knew already that, that myeloid cells were abnormal in lupus patients and that they were increased in the kidneys of lupus patients, and they were associated with a worse prognosis. And we knew from our microarray data that as the mice became sick, the macrophages took on an alternatively activated or reparative phenotype. And then other people have published that dendritic cells come into the kidney, especially in late disease, and that a particular type of dendritic cell, the dendritic cell 2B, seems to be a pathogenic type of dendritic cell. So, but we really didn't understand a lot about the heterogeneity of these cells, or how each cell type associated with outcome, where each cell type was sitting, what the function was, what the origins were, how differentiation of each cell subtype was regulated, and whether we could then use this information to find biomarkers or to alter the function of the pathogenic cells and retaining those ones that were protective. So we, parallel to the AMP, started doing single cell analysis of myeloid cells from 4 different mouse models. And we chose 2 that express TLR7, 2 copies of TLR7, one with fairly indolent disease, that's the SLE1YAA, and one with very aggressive disease, the NZW-BXSB. And then we chose 2 that we call lymphoid expansion, basically because they have expansion of their T and B cells as one of their primary problems that cause their lupus, and they have normal expression of toll-like receptors. So we did dissociation of whole kidneys, we enriched for immune cells, and then we did our single cell analyses. And I'm just going to show you the myeloid cell data. We were able to define 10 different subsets of myeloid cells, and they're shown here. So we have our resident macrophages in the middle here. We have all our dendritic cell subsets over on the right side. So we have DC1s and DC2s. We have these CCR7 DCs that have been shown in lots of tissues, plasmacytoid dendritic cells, and a small subpopulation of myeloid-derived suppressor cells. And on the left side, we have our monocyte-derived cells. So these we divided into classical 1 and classical 2 monocytes, and then non-classical 1 and non-classical 2 monocytes. Okay, and when we cluster these, what we find is that, I'm sorry, the SLE1 label has fallen off here, that they all look fairly similar. And so these are the clusters. I've labeled them here on just this one here from the SLE1 YAA. And you can see to varying extents, they all have the same subtypes. There were more of these non-classical 2 cells in the 2 models with the TLR7 overexpression, but otherwise they look quite similar. And so we were able to make a cluster analysis in which we used all the cells from all the mice to make a single t-SNE plot. Okay, so now if we look at what happened over time in the mice, and we took mice before the onset of their nephritis when they were young, and then we took mice with established nephritis for more than 2 weeks, and we compared what happened. And now we're looking at the frequency of all of these different subtypes in the mice. So what we can see is a number of things that I want to point out. The first is that if we look at these classical 2 monocytes here in yellow, they were very infrequent in the young mice, but they expanded in the sick mice in all 4 strains of mice. And so this was a subset that we were kind of interested in. There was a big influx of non-classical monocytes in the sick mice. You can see there's very few in the young. There's a big influx of them in the sick mice. Resident macrophages, just as a percentage of cells, went down because all of these other cells started to infiltrate. And then we did start to see a little bit of DCs, but it wasn't huge in terms of the dendritic cells. So we then looked at all of the markers of all of these cell types, and we designed a new flow cytometry assay with 16 colors that differentiated all these subtypes from each other. And we were able to optimize a very nice panel that differentiates them from each other. So what you can see here on the left is the young mouse, and on the right is the sick mouse. So the first thing that was very noticeable was that the resident macrophages shown in red had a big phenotypic shift, even with just the 16 colors, comparing the young with the sick mice. So you can see that this subset here is a new subset of resident macrophages that arises in the sick mice. And we also saw these classical 2 macrophages here, as well as these non-classical macrophages over here. Okay, so were we able to see this in humans? So we took our single cell data from the mice, shown here, and again, using Harmony and the help of Shoma Roy-Chowdhury, we overlapped it with the data from the AMP. And in fact, we can see all the same subsets in the humans that we see in the mice without even clustering the human cells. I want to concentrate really on these 2 subsets of cells, the classical 2 and the resident macrophages. So we looked to see whether they were there in the blood or just in the kidney using our flow cytometry assay. So if we look at our 4 subsets of monocytes, and in red, I'm showing you blood, and in blue, I'm showing you kidney, and then the paired bars show preclinical versus clinical nephritis. What we can see is in the blood, starting with the red, the classical monocytes go down and the non-classical ones go up as the mice get sick. They have a lot of them in the blood. If we look at classical 2 in the blood, there starts to be a very small number of them in the blood as the mice get sick. But if we look in the kidney, what we can see is that the classical macrophages actually go down in the kidney with nephritis, and the classical 2 macrophages increase. And then the non-classical ones come in, as I had shown you previously. And when we look to see what was related to what, the yellow is the classical macrophages. So this is done by a trajectory type of analysis. We can see these classical 2 macrophages are very closely related to classical 1 macrophages and non-classical 1 macrophages. Okay, we then, with the help of collaborators in Israel, have done a transcription factor analysis to try and understand what might be driving this change from classical 1 to classical 2 macrophages. And we can see classical 1 is in orange and classical 2 is in yellow, that a certain set of transcription factors was downregulated in classical 2 and some were upregulated. And I want to point out STAT1 that was upregulated in the classical 2 macrophages, and this work is still in process. Okay, so now looking at humans, we knew we could see them in humans, and so we looked at our data. So these are the lupus patients, and these are the healthy donors, and you can see they're there in the lupus patients. They're barely there in the healthy donors, and this is where they're located. And if we looked for correlations with activity and chronicity index, they correlated very tightly with the activity index. And we had 123 patients where we had enough data to do this. They correlated tightly with activity index, whereas classical 1 macrophages and non-classical 1 macrophages were inversely correlated with activity index. So it looks as if these macrophages come into the kidney either as classical 1 or non-classical 1. They encounter damage in the kidney. They differentiate into classical 2, and this is associated with the histologic activity index. And I'm just reminding you that these are located in glomeruli. So what are these cells? Well, there's been a lot of data recently on this cell type in fat tissue, liver tissue, lung tissue, brain, and everyone has a similar phenotype, but nobody's 100% certain whether these are good or bad. I don't have time to discuss all of it. Maybe if we have time for questions, we could talk about it. They have both good and bad features associated with them. Okay, so in the last couple of minutes, I'm just going to show you the data from the resident macrophages. So we subclustered these and we found 5 subclusters of them. And what you can see in all of our models actually is that this cluster RM1 is the dominant cluster in the young and not sick mice. And it gets dominated by this new cluster RM0 in the sick mice. So RM1 goes away and RM0 becomes the dominant cluster. Again, we've done a transcription factor analysis. This has been quite interesting because RM0, which is the one in the sick mice, downregulates FOS and Jun and upregulates or maintains expression of MAF. Where are these cells? Again, in the humans, I'm showing you where they're located. They're located in 2 clusters, and these clusters are separated by expression of the marker LIVE1. These are LIVE1 positive, these are LIVE1 negative, but RM0 and RM1 are agnostic to LIVE1. And you can see that RM0 is spreading away from RM1, that's down here, and it's spreading up here to the quadrant where the classical 2 macrophages are located. Again, just to remind you that they're in the interstitium, and it turns out that RM0, which is the one that is found in the lupus nephritis, increases with disease activity, and RM1 decreases with disease chronicity. So it looks as if the first thing that happens is that you get these RM0s, and then you start losing your RM1s. Okay, so I'm going to just conclude from here. So these mouse models have allowed us to look for novel subsets of myeloid cells in humans, and the association of the classical 2 macrophages with disease activity suggests they're a marker of glomerular injury, although whether they have good or bad function remains to be determined. We've found an unappreciated diversity of resident macrophages, which we didn't detect just looking at the AMP data alone. And so understanding the drivers of these cell states allows for functional experiments in the mouse models. These are some of our future directions, using the mouse cells for co-cultures, using some knockout libraries, looking at what happens with treatment, and starting to look at urine. And I just want to acknowledge my graduate student, Chirag Raparia, who's done the flow cytometry, a lot of the kidney and mouse work in the lab, and collaborators from the Broad Institute, Paul, Nir, and Arnon, as well as funding sources.
Okay.
I'm going to unshare. Okay, there we go.
Good. Well, thanks very much, and fabulous talk, incredible work, groundbreaking. So can you see the questions there?
I can't. Oh, yes, I probably can. Here we go.
Okay, so leave it to you to see. We probably could go a couple of minutes over, so just pick your questions that you want to address.
Okay, so Tim, okay, so racial background. We're starting to look at the APOL1 polymorphism, but part of the problem is that we don't really have enough patients with the polymorphism, with the bad polymorphism. We only have, I think, 60-something African-American patients and 15 with the polymorphism. And so it's been a little bit difficult. That work's being done by Jill Byon's lab, so you have to just keep tuned for that. Okay, so Tim, proteinuria and chronicity. I don't know the answer. Proteinuria is supposed to be bad for you. We didn't look at the amount of proteinuria as in the CNA. It's not one of the things we looked at. It was really proteinuria plus minus, or it was corrected for proteinuria. It's certainly something that people who will have access to the AMP data will be able to look at to see if the degree of proteinuria is worse. And all I can say about other diseases is that classical 2 macrophages are not found in either hypertensive or diabetic nephropathy. We have data from the KPMP. It's very striking. Paul Hoover did the comparison. They do not have glomerular macrophages, and they do not have classical macrophages. We have classical 2 macrophages. We haven't looked at the resident ones yet. And then urine, yes, we can find classical 2 macrophages in the urine of the patients. And so one of our questions is, can you detect them before the onset of nephritis? Can you use them to follow disease? This will require cohorts of patients, but we have matched urine, kidneys, and blood from all these patients. The AMP has still not done all of those correlations.
So, and when you look at the possible interaction between tubular injury and the the immune cell infiltrate. Is there anything you can say about that?
The answer is yes in mice, where it's been looked at fairly extensively, that tubules secrete pro-inflammatory mediators to recruit inflammatory cells, that inflammatory cells are pro-fibrotic, that tubular cells can release things like TGF-beta that activate the resident macrophage cells to become more pro-fibrotic, and there is a lot of interaction there. Within the glomeruli, there's been less done, I think, for obvious reasons, that it's so difficult to deal with, small numbers of glomerular cells. So, we don't know, but it's one of the things that we'd like to look at a bit more in the mouse models.
Okay, maybe we have time for one more question. I think there's one more in the chat there.
Oh.
About existing medications.
Yeah, okay. Yeah, so that's a really good question. And we have some very preliminary data in the NZM 2410 model that we've given BAFF inhibition that that myeloid cells still accumulate in that model, but not all the myeloid subsets accumulate. And so we have ongoing experiments going now where we're going back to that model and we're doing the single cell analyses of those mice after treatment to understand in the setting of a mouse that's still got a lot of immune infiltrates but isn't sick, which cells are there and which cells are missing.
Okay, I think we probably have to leave it there, but thanks again for this really tremendously interesting talk. And I think you're going to meet with a couple of people in the division during the day. So we'll see you later.
Yes, okay. Thanks very much.
Thank you.
I'll put answers in the chat here for the last question.
Yeah. Okay. Bye-bye.